Well, good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Deep Phenotyping of the Human Liver for Non-Alcoholic Fatty Liver Disease, NASH Target Discovery. My name is Samaya, and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Lexogen, who developed the content for this presentation. Lexogen is a next-generation sequencing and transcriptomics company. As the RNA experts, they focus on empowering scientists with innovative, top-quality RNA analysis solutions and support in order to improve health and well-being for everyone on the planet. The product portfolio of Lexogen includes innovative kits for single-cell as well as bulk Rna-Seq, RNA purification and ribosomal RNA depletion, spike in RNA variant controls, and metabolic RNA labeling. Lexogen also provides top-class, fully integrated RNA-Seq service workflows, including sample preparation, multiplex sequencing, data analysis, and report generation. The company headquarters is based in Vienna, Austria, and the North American office is in Greenland, New Hampshire, USA. Now, it's my pleasure to introduce our speakers for today's event. Quin Wills is an MD with further degrees in genetics, mathematics, and computational biology, and a doctorate in systems genomics. He founded his first liver drug genomics company 15 years ago, has steered research consortia that use genomic tools such as single-cell sequencing, and most recently founded the Advanced Genomics Department for leading pharma before returning to the world of biotech with Ochre Bio. Lucas Paul holds a PhD in biomolecular sciences from the University of Manchester and has researched several aspects of RNA biology before developing RNA-Seq kits, services, and scientific affairs at Lexogen. In his current position, Lucas Paul manages Lexogen's product portfolio part that includes services, external controls, metabolic labeling, data analysis, and custom design. So now, without further ado, I'd like to hand the mic to our speakers. So Lucas, you may begin when ready.
All right. Well, everybody hear me? Thanks, Sumaya, for the intro, and thank you everybody for joining. A bit of housekeeping, if I might, just before I begin. You'll see the QR code towards the bottom left corner of the slide. I'll have this up again at the end of the presentation. So if you hear anything of interest today, feel free just to point your phone camera at the code to request the slides or just chat with our team. Right. As mentioned, my name is Quin Wills. I'm the co-founder and CSO of Ochre Bio, an RNA therapeutics company that has evolved out of about 15 plus years now in liver genomics experience in drug development. What I'd like to do over the next 30 or so minutes is walk you through how we're applying deep phenotyping to develop RNA therapies for liver targets of chronic diseases. All in all, there are three take-home themes I'd like to cover. First, I'll chat about what deep phenotyping is and where it fits into the current genetics and genomics trends within drug discovery. Next, I'll provide one example of how we've done this by spatially sequencing 1,000 donor livers together with histopathology machine learning. I'll then finish off with the sometimes prickly topic of machine learning in big data and our goal to build an actionable in silico liver. There's been a fair share of sometimes overhyped ideas since the beginnings of computational biology on how to build in silico organ models. I believe that cellular genomics will start realizing some of these ideas, and I'll share our plans starting with deep phenotyping to generate the data through to the computational models that we believe will reduce our reliance on brute force automation in our space. But before I get into all of that, I'd like to describe the human problem that has us thinking about these themes, starting with perhaps a sensitive rhetorical question. Who of us here have gained a few COVID kilos, right? Weight has become one of the most discussed topics during the COVID pandemic, especially as almost 2/3 of us entered the pandemic with a weight issue now recognized as a risk factor for poor COVID outcomes. But we've always known that getting older and gaining weight are a toxic public health combination. Just a year before this pandemic, Dr. Ritchie of the Oxford Martin School put out this wonderful health news piece explaining global mortality statistics and trends. I quite like this graph because it amazes me how few people are able to guess what kills us. Cardiometabolic disease, in other words, cardiovascular diseases and diabetes on this chart, are twice as likely to kill you as all of the cancers combined. I would go a step further to argue that this is probably an underestimate, as we know that obesity plays a causal role in many of these disease endpoints. So put simply, your aging metabolism combined with a calorie-rich sedentary lifestyle will likely be a significant factor in your undoing, and no number of new blood sugar, blood lipid, or blood pressure medications seem to be turning the tide as we live longer as a species. 1 in 3 women now born in the UK can expect to live beyond 100. And it's frankly just not going to be effective to tell these individuals that they need to focus on eating less and moving more so that they can be healthy, productive 100-year-olds. We need better cardiometabolic therapies for the last 30 to 40 years of life. We need more actionable data on the many facets of aging metabolic health in our modern lifestyles, such as the biology behind this statistic. 1 in 4 of us have harmful levels of liver fat, and that number is rising fast. As you gain weight with age, you not only build up fat around your internal organs, some organs such as the liver start storing excess fat. We now know that this fat inside your liver cells is a major player in cardiometabolic diseases. We also now recognize that this excess liver fat is becoming the leading cause of needing a liver transplant. Sadly, too much fat in the liver is also becoming a major reason for the shortage of donor livers, as fatty livers don't transplant very well. What this means is that despite significant advances in liver transplant medicine, we are transplanting poorer quality livers, with many patients dying on the transplant waiting list because of short supply. You might have heard of this disease under many acronyms. I'll refer to it in this presentation as Nafld, which stands for non-alcoholic fatty liver disease, but some call it Mafld to stress the metabolic nature. You might also hear of NAFL for early disease and NASH for later disease when the liver inflammation begins. Here's a simplified model of its progression with the systemic and liver-specific consequences. Currently, if you develop cirrhosis, there is no going back. The only cure is a transplant. However, the problem with transplantation for Nafld is not only the short supply of good quality donor livers, as mentioned a moment ago, but also a risk of disease recurrence that is greater than 50% within a year. If we hold off on transplants for alcoholic liver disease unless adequately managed, should we be providing transplants to obese Nafld patients in the absence of meaningful post-transplant Nafld therapies? Ochre Bio was founded to develop therapies for all of these challenges. We began with a mission that we should squarely address the 2 biggest translational challenges that face Nafld. These are challenges we believe that are shared with many chronic diseases. The first challenge is that Nafld is a silent disease. Even most patients with cirrhosis won't know they have it until it is so severe that the liver cannot compensate for the extensive scarring and lack of functioning liver cells. This means we have very little data on the disease's progression. It means we have experimental models of unclear relevance for identifying targets. It also means we don't have validated biomarkers for clinical trials. So it's no surprise that Nafld clinical trials are experiencing exceptionally high failure rates. The second challenge faced is that Nafld is complex. Not only does it have many cardiometabolic interactions, but the more the disease progresses, the more cell types and pathways become key players in its pathogenesis. And so the less likely it becomes that a single therapeutic target will be effective. It's these two challenges that are the motivation behind the take-home themes I'm discussing today. We want to, one, learn to break the silence of this disease, and two, understand how to treat it at any stage of complexity. For us at Ochre Bio, we're starting with the following. This is a donor liver being kept alive on a machine with blood and nutrients. Recent breakthroughs in machine perfusion are not only allowing surgeons to better assess donor livers, but also offer the opportunity to regenerate suboptimal donor livers before being transplanted. Our goal is that in the near future, every suboptimal liver on a machine will receive one of our long-acting gene silencing RNA therapies. This will increase the supply of donor livers, improve transplant outcomes, and crucially reduce Nafld recurrence. As you can imagine, studying discarded fatty donor livers on these machines is also proving to be an incredible research model. So our vision is ultimately that nobody should die waiting for a liver, and nobody should receive a liver that won't stay healthy for the rest of their lives. More so, no one should live with the complications of Nafld in the first place. By starting with the first two, our belief is that we will more effectively develop therapies for patients chronically living with Nafld. And I'm sure you've guessed by now, that journey for us begins by generating the type of data that we call deep phenotyping. So what is it really? I like to begin by pointing out how population genetics is increasingly being used in drug development to connect one or more genes to disease. Genetic health data helps de-risk targets by front-loading causal information. The problem is, this is all that population genetics does. It doesn't provide any of the biological details that are so crucial to understanding chronic diseases like Nafld. Details such as which organ the gene plays a role in, at what stage of the disease, or what the mechanistic biology is that the gene is part of. So that's where deep phenotyping comes in. From a technology point of view, one could describe deep phenotyping as the marriage of genetics, imaging, cellular genomic methods like single-cell or spatial sequencing, and then statistical or machine learning to model the data. It's the generation of rich tissue-level information mapped to genes and clinical phenotypes to fill in those gaps. And here's a real-world example of a deep phenotyping project that we recently completed to better understand those silent years of Nafld. In partnership with transplant surgeons at the University of Oxford, we studied 1,000 donor livers. Each liver was imaged at least 6 times for histology. Here's an image containing biopsies from 2 of the livers. The white holes you see are the excess fat stored in the liver cells. Histopathology machine learning not only scored the histopathological features for us, but was trained to identify tissue niches of particular interest. For example, the automatically identified areas in blue here contain cells undergoing what is called ballooning degeneration. As you might imagine, this richness of histopathological data in and of itself allowed us to ask detailed questions about disease progression. But we wanted to take it to the next level and overlay this imaging with spatial RNA sequencing. As many of us, I'm sure, now know, spatial genomics has attracted some attention over the last years, recently winning the Nature Journal's Method of the Year. It's the newest member of the cellular genomics toolkit and faces somewhat similar challenges to single-cell sequencing, not least of which is sample processing. In this case, we needed a method that could work with extremely challenging biobanked Ffpe samples, something not yet generally supported with commercially available platforms. So we decided to come up with our own solution. Working closely with Lexogen and QuantumSight, we converted this niche imaging data into masks that were printed over the tissues, covering all areas except for the niches identified here in blue. Each one of these niches was then lysed for RNA sequencing using a protocol we developed with Lexogen. This then allowed us to ask 3 types of questions. How more disease niches compare against less disease niches, and how much of this is dependent or independent of the tissue microenvironment. How niches compare across donors, and finally, how the gene expression changes map to 150 clinical variables, from blood proteins, lipids, and metabolites to donor age, obesity, and medical history. Here's an example output of some primary analysis in our Lens software. This is a gene that is part of a gene family implicated in Nafld, but which itself has not been associated with the disease. On the left, we present its hub of closely correlating or anti-correlating genes as a simple plot to explore. The yellow bars are genes of particular interest to us, capturing 2 areas of biology: tissue regeneration and nutrient sensing. This already gives us a clue into the pleiotropic biologies associated with this gene. The right-hand side plot is of its associations with clinical and histological phenotypes. The x-axis maps the log-scale p-values using marginal linear statistical models, while the y-axis maps how important each gene is in a multivariate nonlinear machine learner. I've annotated a few here that we consider relevant, from associations with cardiac disease to percentage fat in the liver. Here's an example of output from our secondary models in our engine software, which I show here to demonstrate the validity of this type of data. Our engine software contains over 250,000 data sets that help us contextualize this type of data. Here I've asked the software which of the data sets most closely resemble the signature of genes associated with percentage fat in these livers. It presents the results as a network that it also clusters. Each point here is one data set. The main cluster of data sets I'd like you to focus on is the purple cluster, where the top 10 hits also have a black point in their centers. I've annotated 9 of these, being phenotypes from the study that are tightly correlated with percentage fat. What's interesting is the 10th point. It's a gene signature extracted from a published study of liver fat accumulation. What's more is that many of the points in this cluster are also studies of liver fat accumulation. So in this atlas, we are seeing what others see, but more, not only because of the statistical power that such a study affords, but also because we use spatial information to find genes others might overlook. Here's an example of another such gene. This is a gene that was top ranked when we asked a many-to-many machine learner which genes predict histological disease progression independent of obesity and age, which are the major clinical risk factors. Looking at 3 examples of histological associations here, we see along the y-axis that it is positively associated with key disease histological phenotypes. Clinically, it demonstrates a positive association with both girth and blood gamma GT, which is a measure of liver damage, and this makes it particularly interesting. However, it's the bottom right inverse association with C-reactive protein, which is a measure of inflammation, that really caught us off guard. That was until we broke down the data spatially, which I'll show here with one simple plot. Here we break up the gene's expression into healthy tissue niches, shown in green, fatty tissue niches, shown in yellow, and niches beginning to show degeneration and inflammation, shown in red. In the latter niches, we observe the same reverse association with inflammation, which I show here as immune cell count. This turns out to be an absolutely fascinating gene that we are studying in the lab, and which we believe could play an important therapeutic role in managing the liver-specific complications of Nafld. Now, I mentioned at the start that this type of data generation is only the beginning of the process towards building actionable models. It's a process that we've come to call our 5 Cs. What I've been describing up to this point is our charting step, the curation and generation of rich data that allows target discovery. The next 3 Cs are what happens in our Oxford lab and maps roughly to what many target validation labs might do. It's to clearly establish a gene's causality within one or more stages of disease, understand its complexity, i.e. the genes it interacts with and the many effects it may have, and its coherence, which in the simplest case means it produces the same result in multiple models. 2 key differences though between this approach and perhaps many target validation labs is that we only work in primary human cells and tissues, and we sequence everything so that we can more deeply understand the underlying biologies. So deep phenotyping doesn't end with charting, but it continues all the way through the pipeline. Where we also differ from traditional pipelines is that we don't use animal models to validate the biology perturbed by our lead candidates. We go on to confirm our therapeutic efficacy in perfused human livers, similar to that shown in the video. Again, all the while we sequence, so that ultimately we can compare all of the data to the greater body of data that we generate, including benchmark genes and probes. So one question is, how much deep phenotyping and how many iterations through this 5C process are really necessary? Well, that depends on the questions being asked. Recently, we opened our Taiwanese lab to augment our deep phenotyping from 250,000 datasets to over 10 million datasets. We're doing that using single-cell sequencing so that we can now better study all of the cell types in the liver. Here is a plot of tens of thousands of liver cells. To date, we have exclusively focused on the green group of cells in the bottom left corner. That is because we use gene silencing RNAs called siRNAs that we can target specifically to these cells using a sugar ligand called GalNAc. Together with RNA chemical modifications, we can shut down genes in these cells for months at a time. After decades of research, RNA therapeutics have finally reached broader medical practice, with Alnylam's siRNA that models liver cholesterol homeostasis, and Moderna and BioNTech's explosion into mRNA COVID vaccines. In our world of liver deep phenotyping, RNA therapeutics offer significant advantages. We're able to progress very rapidly from target to therapeutic, and it makes it easier to consider advanced therapies like combination therapies, where we can modulate duration of effect. The challenge, however, remains targeting specific cell types. If we are to reach our full potential of regenerating livers at all stages of disease, we will need to understand and target these major groups of cells too. As you might imagine, it's work that requires more than just generating a lot of data. In this case, novel chemistry to develop ligands that target other cells, but more broadly, computational modeling and machine learning to turn the data of the interacting cells into actionable insights. Which brings me onto that final theme of this in silico liver. We often get asked if we're trying to be the Google Maps of the human liver. I like this analogy because Google Maps is based on a huge amount of data. Many of us have seen the Google cars with cameras driving around cities capturing this information. More importantly though, Google Maps has many sophisticated models based on the data that make real-time predictions for you, like what the fastest route will be. This is exactly what we're trying to do, not just gather rich data, but turn it into good predictions. In particular, We are using computational models to help us solve an age-old technical issue in biological science. Sidney Brenner once pointed out that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. Many of us would definitely agree. New technologies really drive what we can do in biology. The problem is that classical cell and molecular biology techniques fall into the bottom left part of this plot. They don't scale very well and typically make simplistic, isolated measurements. With the shift in drug discovery from chemistry-led to biology-led innovation, we are seeing a massive push for brute force automation and scalable in vitro models. The problem is that this often restricts us to make simple measurements in overly simplistic models. So we fail cheaper and fail faster, but don't necessarily fail less. Deep phenotyping helps us to fail less using content-rich measurements in tissues, but this often comes at a price of scale. We want to get here, and to do that, we need computation. And that's what we mean by an in silico liver, predictive models that strike this right balance between throughput and content. So fail cheap, fail faster, but equally crucially, fail less. And here's how we're doing it. Every week, we receive biopsies from patients with Nafld. That's what the white arrow top left is pointing at. These biopsies are on a machine that turns them into very thin slices that we can culture in the lab, as shown in the plate bottom left. Our lab in Taiwan has been established to single cell sequence these slices, where all cell types in each slice have had one or more genes knocked down. We call this approach phenotype-free screening because we're not biasing ourselves to any one measure, but are sequencing every gene in every cell in response to gene perturbations. It's this data, together with all other data we've generated to date, that feeds into this in silico liver. I've shown you how we've used machine learning to automate histopathology or provide a more nonlinear framework for finding genes that predict phenotypes. Here we are using machine learning to run in silico screens so that we are not overly reliant on brute force automation, focusing rather on quick iterations in high quality human tissue models where we have the option to make more complex measurements. On the right hand side is a very simple plot that explains conceptually how we do this. Let's imagine that we want to study which genes modify a clinically relevant phenotype in these liver slices. Let's say cell death, for example, in response to the kind of ischemia livers face during transplantation. The y-axis is the measurement of the phenotype, where each point here is how that phenotype changes when a gene is knocked down. Let's say we have another measurement that has some relationship to this phenotype of interest. Here we call it the explanatory variable and place it on the x-axis. We can use the explanatory variable to predict the phenotype of interest. Now let's imagine that we have over 10 million explanatory variables thanks to deep phenotyping, and that all of these have been measured against every gene. That means two things. One, we can build a very rich model of what explanatory variables predict the phenotype. I'm showing this as a simple curve here for easy visualization. Two, we can make predictions for genes not tested against this phenotype, which I'm showing you here as solid points. So our aim is that by next year, we'll have reached a point where we're able to perturb no more than a couple hundred genes to study any complex phenotype of interest in liver slices, and that the in silico liver will predict the effects of all other genes in all cells, while also giving us mechanistic insights into what might be good combinations to target. This is how we believe deep phenotyping plus in silico modeling can complement each other to study liver biology at scale without always having to resort to hefty studies that can only be properly addressed with brute force automation. We want the option of quick studies that allow us to rapidly iterate through hypotheses using better measurements in better models that capture the complex biologies in aging and obesity. Which brings us to the final slide and back to the human problem that fundamentally motivates us. Our aging population, with the spiraling healthcare costs shown here in our latter years of life, needs to shift away from only treating end-stage complications towards also treating the chronic, silent, and complex fundamentals fueling these diseases. We believe that one of the best ways to flatten this curve is by deep phenotyping and ultimately correcting the cardiometabolic fundamentals in the latter 30 to 40 years of life. And honestly, we believe there is possibly no better organ to do it in than the brain of your metabolism, which is your liver. So with that, I'll stop. Thank you, everybody. I'll quickly sign off with a big thanks to the Oxford transplant scientists, the Birmingham transplant scientists, Lexogen, our technology partners in California, and the many others all around the world who have been and continue to be a part of this really fascinating journey. Thanks, everyone.
Great, thank you, Quin. Now we'll be turning to Lucas Paul from Lexogen.
All right, thanks, Sumaya. And thank you, Quin, for this very impressive presentation. And we at Lexogen, we're really happy to work together with you on this transcriptome analysis. And that's actually what we do at Lexogen. We are RNA experts and we specialize in transcriptome sequencing. Geographically, we are located in Austria and we have a subsidiary in the US, but of course we serve our customers worldwide. And we have a very comprehensive product portfolio across the whole RNA sequencing workflow. So from sampling to RNA preparation and NGS library prep. What we've seen today was gene expression profiling, but we can of course also analyze whole transcriptomes, small RNAs, and so on. And yeah, just to remark on this current crisis, a lot of our products are used in COVID research, and of course we also have a SaaS panel based on RNA sequencing available as well. Now, a typical Rna-Seq service starts with a consultation, like it was in this project, that we have to learn about what our customer really wants, what it needs, what are the specialties here. We have to look into the sample source and the input, which was very particular here. This was liver biopsies, Ffpe samples, very low input. So we decided on the correct library preparation here. The sequencing then occurs on the Illumina platforms with a read mode and read length that is suitable here, of course. And then we can provide data analysis and also comprehensive data analysis on other platforms as well if needed. So this means we are a special Rna-Seq provider. We can ensure a personalized treatment, and we are producing and developing our own kits, which can give you the peace of mind that we have the technical expertise also to tackle very challenging samples, meaning degraded RNA input, meaning really low input samples. And of course, we have the turnaround time to match these promises as well. And in terms of applications, that means that we cover gene expression profiling with our Quant-seq method, which is a 3' sequencing method for tag profiling. We have a version of this called Quant-seq pool, which was applied here that takes care of especially samples that are very homogeneous in quality and amount. And of course, we can also look at the whole RNA from the 5' end to the 3' end for SNP detection, for isoform variant detection, and also can look for non-coding RNAs using our ribo depletion kit. We look at the regulatory RNAs as well using our small RNA sequencing approach. And with our latest development, we can now tackle also single cell sequencing and ultra-low RNA sequencing in the picogram range. So I'll finish this with just introducing our springtime offer. Please get in contact with us if you're interested in expert RNA sequencing services, and you can reach us at lexogen.com or at services@lexogen.com. And with this brief introduction, I would like to return the control back to you, Sumaya, and thank you.
Wonderful. Thank you very much for that insightful presentation. Now, I would like to invite our audience members to continue sending in their questions or comments using the questions window for the Q&A portion of this webinar. I've already received some questions, so I'll start off with those. And our first question is, if Nafld is a disease of obesity, why not just focus on weight loss?
Um, I think if we are to if we're even to ignore the sort of later stages of disease and the cirrhotic implications, there are really two answers to this. And the one is, we know that the liver isn't just a victim of what's happening here. There really is a two-way process. The liver talks to the rest of the body in adipose and the other way around. So there definitely is a very strong argument for targeting the liver in this disease. But more broadly, I would just say that increasingly we're realizing that it isn't good enough just to have this idea of we need to tell people to move more, eat less. We do need to start thinking of obesity as a disease in and of itself to be treated and focus on that.
Great, thank you, Quinn. And our next question is, your strategy seems a lot like longevity medicine. Is that the goal?
We definitely target many pathways that are of interest in longevity medicine, but that is not explicitly our goal. Our goal is really that final slide that I presented. We want to help people live healthier longer. I want to be a 100-year-old that is, still climbing mountains and annoying my team for the latest data. I don't want to be sitting staring out a window for the latter years of my life, and that's really what we're trying to get at.
Great, thank you, Quinn. And our next question is, with all of this sequencing, are you using biomarkers and stratifying patients?
I think the best way to answer that question is to say, at the end of the day, we're focused on impact, right? And of course, we're generating a lot of data, and as scientists, we are very curious about stratifiers and looking for biomarkers that we could take forward in trials. But we don't want to be limited by these. And so that was the founding thesis as a company, that we want to be able to maximize our impact in the NASH space, and we want to do that by focusing first on the transplant space so that we don't have the same biomarker issues that we believe are really pervasive in the NASH clinical trial space. In terms of stratifiers, it's the exact same thing. We want to maximize impact, and sometimes that is by targets that have a broad application. But if we see targets that have a very specific application, so lower reach, so a smaller population, but have a much bigger impact, definitely that is something we'll go for.
Wonderful. And another question from an audience member, and that question is studying human livers on machines a good model?
It's not a perfect model. But really in science, we accept that all models are wrong. It's just that some are useful. So when we talk about utility, what do we mean? And really, utility is two things in the modeling space. It's something that allows you to make predictions and or something that allows you to understand what's going on. And so in our experience, when we've looked at models in this space, things that have failed to tick those boxes are cell lines. So cell lines as models are really poor in the space for making predictions and helping us understand the complex biology. I would even say iPS models, so stem cell models at this stage in liver biology, are also not great, partly because they behave like young liver cells. Animal models are not great. So for us, really, the focus has been we have to work with human tissues. And so we work with human tissues in the lab and ultimately work with livers on the machines. The proof in the pudding, again, will though be getting things to clinic, right?
Great. And another interesting question from an audience member, and that question is, it's not clear to me why you don't just immediately target patients with NASH. Could you explain?
You know, if you think back to the first few slides when I mentioned the two big challenges around Nafld and NASH, you know, these were the things that we thought long and hard about as a group. And the first one was that one about silence. You know, the disease is silent for so many years. That means several things, and one thing it definitely means is that we don't really have good biomarkers. And for us, if you don't have good biomarkers, you are always going to struggle with clinical trials. And so we wanted to think about a way we can take on NASH without having to do NASH clinical trials. And so the idea then was to focus on transplants, where in the transplant space, there are well-established biomarkers to understand how well the liver is doing once it's been transplanted.
Great. And another question from an audience member, and that question is, what were the biggest technical challenges sequencing very small spots of tissue from FFPE slides?
Oh, I'll hand that one to Lucas.
I'm happy to take that one. So I have to say that was a particular service. So our technology, our quant-seq technology, is focusing on the 3' end, which means that it can cope with degraded RNA samples and small RNA amounts. So the RNA extracted from these biopsy samples and FFPE samples was, of course, of lower complexity than fresh frozen samples, but our technology can genotype this and get the gene expression profile to the spot. And I just want to mention that we also provide spiking controls here that give a measure of the performance of the whole procedure, and that allows you also then to decide which data sets to compare. So that is something that we have in addition to the usual service because we develop the controls ourselves as well.
Thank you, Lucas. And another question from an audience member, and that question is, do you know what changes occur in liver at gene level with metformin therapy?
Yeah, the magic of metformins. Well, you know, Metformin is still slightly a mystery, right? We know the effects on AMPK, and we know we have some ideas on the cascade that plays out. I think one of the really interesting questions in the liver space is, does metformin really improve insulin sensitivity, or at least improve insulin sensitivity together with a reduction in steatosis? And I think the evidence is not quite there yet. When we look at things at a gene expression level, with a lot of the signaling that plays out, we don't always see very direct signals of, for example, AMPK activation. But what we can see is some of the stuff around IGF signaling. And we definitely see some of that playing out when we spike metformin into models.
Thank you, Quinn. And another question from an audience member, and that question is, how many different liver biopsies are tested with the gene silencing assay? Individual livers may have mutations or different regulatory alleles that may give different results from a normal liver.
Yes, so when we are using livers as models, so when we're making these precision slices, the golden rule is you check when you do a gene knockout, you do it across many livers, because it's very difficult to know how each liver will respond and how much of that is innate, so genetic, and how much of that is a factor of the quality of liver that you received. And really it boils down to just doing it over and over and over again to make sure you have that result.
Thank you, Quinn. And another question from an audience member, and the question is, did you use genetic data?
We always use genetic data in our work. So for example, the UK Biobank association results, we use that. It's in our engine software. We refer to it all the time because it really, again, helps front load causality in our models and thinking about which genes we want to chase. We didn't use genetic data in this study. We weren't sufficiently powered to say anything genetic. with this study. But what we will be doing with the atlas that we're building in Taiwan is exactly what genetic data does, is directly perturbing each gene and seeing what the consequences are.
Great, thank you for that. And another interesting question from an audience member, and that question is, besides spatial technologies, what types of technologies, specifically hardware, should tech developers be focusing on for the future of therapeutics development?
Yeah, wow, that's a very big question. I think, again, this is a lot of the answers to these things are very domain specific, right? So know your domain, know the questions you want to ask, and then know what the options are to take that on. There are a lot of philosophies, and so, for example, we've touched on one of the philosophies today of sort of big automation. Now, I'm not against automation at all. A sort of big automation is a great idea in many spaces, and stem cell models and big imaging to interpret the stem cell models is also a great idea, particularly in spaces like neurodegenerative disease. I would say for us, for example, what is proving to be a lot more useful is focusing on sort of the cellular genomic techniques and being able to do the kind of thing that we're talking about now with this in silico liver. And that really, you would be surprised, doesn't always require the big software that you might need if you're doing big imaging with big sort of big high throughput screens. Everything we run on the cloud with our I suppose I can say with our AWS servers. So it's all very manageable and something that is quite tractable for biotech companies these days.
Wonderful. And another question from an audience member, and that question is, you mentioned both single cell and spatial methods. Can you describe each of the methods you use and why you chose those specific methods? What was the validations process you used to determine best practice for your application?
Oh, how long do you have for that answer? When deciding what technology to use, like I said in the previous question, you always start off with exactly what questions you want to ask. And so, for example, when we started with this project that I talked about today, the 1,000 liver atlas, we were very focused on being able to understand hepatocyte biology. So we didn't necessarily need sort of single cell sequencing to identify each subtype of cell. We were very focused on the ability to be able to study the histopathology and also map it to clinical phenotypes. And to do that, we needed to work with biobanked tissues. And if we were going to work with biobank tissues, the limitation came in that we had to work with FFPE samples. And so from that, we looked at various platforms, and a lot of platforms these days are beginning to support FFPE, but I would argue probably they're still best suited for fresh FFPE, less so sort of biobanked FFPE samples that have been sitting around for a few years. And when we tried things out, we looked at a lot of what we look at in the sequencing space, the quality of the RNA or the cDNA we were getting out of the samples, and what we could do. And that really sort of very quickly distilled it down to this particular solution that we wanted to go for. Will this be the solution that we'll go for maybe in 2 years' time in this space where the technologies evolve very quickly? I can't say. But for us, that was definitely the solution. Now that we're thinking about how to target other cell types and other stages of disease where there is incredible complex interactions of cells, we definitely want to use single cell sequencing. And for that, we need to be able to work with fresh tissues, and working with precision slices is turning out to be a fantastic idea.
Great, thank you, Quinn. And another question, and that question is, how does this type of work overlap with big population genetics initiatives like the UK Biobank?
As I mentioned, we use the UK Biobank data. Again, don't get me wrong, I love population genetics. It's so fantastic to see how genetics is finally coming into drug discovery as another arm in biology to drive innovation. And again, it's doing this by front-loading causal information. And so we use it, and we always keep a keen eye on what else they're doing. But it is population genetics. I think the thing that's quite unique about the UK Biobank is its population genetics that has a strong, deep phenotyping philosophy. What they are doing is generating a lot of phenotypic data around a lot of individuals over time. And some of that data is in fact imaging data of individuals, so imaging of their livers to estimate percentage fat. The key difference again is none of this data really is molecular data in the sense of it gives us a sense of how the genes and proteins in the liver are behaving. And that's where we have to jump in and get on with our work.
Fantastic. And another question, and that question is, what kind of machine learning do you use?
Well, asking about machine learning models, I suppose, is asking a little bit, is like asking about lab models. We use a lot of models, and we use models that work best for certain questions. As I mentioned in my talk, we use machine learning for automation, and a lot of models work very well for that. I think deep learning is very vogue right now, and it works very well for imaging data. We also use machine learning as another form of modeling that sometimes works better with highly nonlinear data. And we have other types of models, type of machine learning models we use for that. And our in silico liver ultimately uses a slightly different version of that. All the time for us, again, it's all about utility. And does it help us make good predictions? And does it help us understand some of the biology underneath that?
Fantastic. Thank you, Quinn. And the next question is, how do you validate your machine learning models?
Well, there are well-established validation methods in machine learning, and really simply what that means is you train the algorithms on a subset of data, and then you test what kind of predictions the algorithms make on a different set of data that it's never seen before. And this will give you some sense of how well the algorithms will perform in the wild, once you set them free. But really for us, again, in many ways, there's no difference between sort of computational models or lab models. The proof in the pudding will be how we get to clinic and how well we perform in clinic.
Our next question is, you mentioned combinations. How predictable are combination effects from single gene knockdown measurements?
Oh, that's a really good question. Well, let's take the question a level up and talk about predicting combinations, right? We all talk about the need for combination therapies. The world needs combination therapies. I think maybe one way to frame this question is to compare thinking about combination therapies in chronic cardiometabolic diseases versus thinking about combination therapies in the cancer space. And there are problems here that we share, and the problems here that we differ on. One area that we overlap a lot in when we're talking about cardiometabolic disease combinations and cancer therapeutics is we all face similar regulatory challenges. And this has been a really interesting learning for us as a company, because as scientists, we have all these great ideas about how we can come up with combinations, and without wanting to attract hate mail from the regulators, a lot of regulation is not yet there. It's not set up to support thinking about clinical trials with combination therapies in a way that would empower developing combination therapies up front. So it comes as, again, no surprise that when you look at clinical trials and combination trials, these are almost always therapies where the individual components of the combination were first trialed in the space. And that's suboptimal. That's not what we'd like to do. And it's not something, unfortunately, that we can solve right now as a company. This is really a regulation thing to be thinking about. In terms of the fundamental biology, I think where we differ in the cardiometabolic space versus the cancer space is that I think increasingly we're appreciating that it's less about saying in cancer, you think about sort of synthetic lethality, trying to find this perfect epistatic combinations, the weakness in the pathway that you can exploit in cancers. And for us, it's more about finding complementary genes. So what I mean by that is genes that act on different pathways at different points in the disease process and in different cells. And that's what you see when we're looking at combination trials playing out at the moment in NASH. For example, the typical combination are therapeutics that are targeting more of the cell stress, the cell death, and the inflammation, combined with therapeutics that are focusing more on the metabolic and obesity elements of the disease. And that is very much how we think about things when we're looking for things, for targets in our data.
Fantastic. Thank you, Quinn. And another question, and that question is, how are you modeling gene interactions to predict combination therapies?
So again, sort of to speak to that, the previous question, really for us, our thinking is more around complementarity rather than maybe a traditional epistatic way of thinking about it. And that is, genes targeting different pathways at different points in the disease process in different cells. And really, to be able to do that in any real way, we do need single cell sequencing. And so that is something, it's definitely going to be a big deliverable from the work we're doing in Taiwan.
Fantastic. Our next question is, will single cell sequencing really reveal enough biology?
Excellent question. Single cell sequencing comes with a lot of compromises, right? As with any technology, it's not true to say single cell sequencing will allow you to see every cell and every gene. You'd never see every cell. We know, coming back to the challenges with processing samples, there will always be processing biases. You won't always see every cell type, but we do our best, and we are at a level, I believe, with liver single cell sequencing that we are seeing the important cell populations to model. We also don't always see every gene, and that's because the compromise we make is by covering lots of cells, we don't sequence as deeply within each individual cell. So you don't see as many genes. But for us, this is less of an issue for the type of in silico liver we're building, because really what we're modeling there are the actual perturbations rather than making a direct comment about any one gene in any one cell.
Great, thank you, Quinn. And we have time for just one more question. And that question is, do you believe that automation kills innovation?
No, I'm really not against automation at all. I just think, and I mentioned this in one or two questions before this, that know the questions you want to ask and know how you can maximize your sort of how quickly you can iterate and sort of move through the space. And my concern is that in biology, in our enthusiasm for big automation, that we're not learning the lessons that chemistry learned. Because a few decades ago, chemistry went through this process with big automation, in the drug discovery space, and some of it was great. A lot of it wasn't, quite frankly. And I worry that we're going to make the same mistakes in biology. Again, for us, it's really about generating that rich kind of data. And if we want to generate that rich kind of data, we're always going to be limited in the type of brute force automation we can use. Our philosophy to this is rather focus on the computation. And if there is no way to address that, then then definitely go for big automation.
Wonderful. Well, thank you very much for all of those answers. We've reached the end of the question and answer portion of this webinar, but not to worry. We did receive lots of questions, so if we couldn't get to all of your questions, not to worry. The team at Lexogen may follow up with you later on as well. And if you do have further questions, you can also direct them to the email addresses on your screen. So go ahead and quickly jot those down. And thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Now, please join us in thanking our speakers, Quin Wills and Lucas Paul, for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.